Hello, everyone, and welcome back to this masterclass in clinical embryology. This channel is designed to give you clear, precise, and exam ready notes in a way that no other channel provides. My goal is that the more you listen, the more you feel glued to this channel and you keep subscribing, watching, and learning from all the content here. Today, we will discuss Chapter 6 The Embryo, from zygote to blastocyst. The human embryo is defined as the developing organism from the time of fertilization up to 8 weeks. Historically, the discovery of the embryo has been one of the greatest milestones in biology. In 1827, Carl Ernst von Baer discovered the mammalian egg. In 1876, Oskar Hertwig described the process of fertilization in sea urchins. In 1978, the first baby was born through assisted reproduction, named Louise Brown. Why do we study embryos? Because this stage of life is the foundation of all organ systems. Understanding embryos helps us in understanding congenital anomalies, miscarriages, and the biology behind reproduction. Fertilization is a sequence of highly coordinated steps. The sperm first undergoes a process called capacitation inside the female tract. This allows it to prepare for fertilization. The acrosome reaction then releases enzymes that help the sperm penetrate the outer covering of the egg, called the zona pellucida. Binding of sperm proteins and egg receptors ensures the correct match between the two gametes. Finally, the sperm fuses with the egg and a cortical reaction occurs, which prevents more than one sperm from entering. The result is the zygote, a diploid cell with 2 sets of chromosomes, one from each parent. At this stage, 2 pronuclei are visible, which embryologists often use to confirm successful fertilization. This zygote is the very first form of human life and has the potential to develop into the complete organism. The zygote does not rest. Within about 24 hours, it starts dividing. This process is called cleavage. On day 1, the zygote divides into 2 cells. By day 2, it becomes 4 cells. By day 3, it becomes 8 cells. And by day 4, it becomes a compact structure called the morula, containing 16 to 32 cells. One important event happens on day 3, compaction. Here, the cells flatten against each other, form tight junctions, and begin to show the first signs of differentiation. In the laboratory, embryologists grade these early embryos based on how symmetrical the cells are, how many fragments are present, and whether the cells are multinucleated. But remember, the details of embryo grading will be taken up in a separate lecture, so you will learn this in depth later. The morula is like a mulberry-shaped cluster of cells. By day 5 or 6, it transforms into the blastocyst. The blastocyst has 3 important parts. The trophectoderm, which will go on to form the placenta. The inner cell mass, which will develop into the fetus itself. The blastocal cavity, a fluid-filled space that gives structure and expansion to the embryo. At this stage, the embryo also hatches out of its surrounding shell, the zona pellucida, which is essential before implantation can occur. This transition from morula to blastocyst is a critical milestone and is often considered a strong predictor of successful development. Around day 15 or 16, another important event occurs: gastrulation. This marks the beginning of body plan formation. A structure called the primitive streak appears, and cells start migrating to form 3 layers. The ectoderm, which forms the skin, the brain, the spinal cord, and nerves. The mesoderm, which forms muscle, bone, blood, and the reproductive and urinary organs. The endoderm, which forms the lining of the gut, the liver, pancreas, and lungs. Along with this, extra embryonic structures like the amnion, yolk sac, and chorion also appear, which are essential for nutrition and protection of the embryo. Around day 15 or 16, Another important event occurs: gastrulation. This marks the beginning of body plan formation. A structure called the primitive streak appears, and cells start migrating to form three layers: the ectoderm, which forms the skin, the brain, the spinal cord, and nerves; the mesoderm, which forms muscle, bone, blood, and the reproductive and urinary organs; the endoderm, which forms the lining of the gut, the liver, pancreas, and lungs. Along with this, extra embryonic structures like the amnion, yolk sac, and chorion also appear, which are essential for nutrition and protection of the embryo. At around day 3, the embryo switches from using maternal RNA to activating its own genome. This is called embryonic genome activation. Several signaling pathways regulate development. WNT helps in forming the body axis. Bone morphogenetic proteins help in forming bones and muscles. Hedgehog signals help in limb and organ development. Fibroblast growth factors help in blood vessels and limb growth. Notch signaling controls how cells decide their fate. Apart from genes, epigenetics also plays a role. DNA methylation and histone modification control which genes are turned on or off. Errors in these processes can cause congenital conditions such as Beckwith-Wiedemann syndrome and Angelman syndrome. The study of the embryo has direct applications in reproductive medicine. Embryos can be assessed morphologically by looking at their shape, number of cells, and degree of fragmentation. Kinetically, by using time-lapse imaging systems that capture every division. Using artificial intelligence, where computer models predict which embryo has the best potential. Embryos can also be preserved by vitrification, which is a rapid freezing technique that is much more successful than the older slow freezing method. So, to summarize, the embryo develops through a precisely timed sequence of stages, from fertilization to cleavage to morula to blastocyst. Implantation is the rate-limiting step for successful continuation of pregnancy. Each stage has clinical importance, and each is carefully studied in the laboratory. Looking into the future, scientists are working on artificial gametes derived from stem cells. Embryo-like structures called blastoids are being created to study development. Organoids are being used to mimic early stages of life, and advanced tools such as artificial intelligence and multiomics will likely change the way we select embryos in the future.
Thank you for watching. If you found this video helpful, please like and subscribe for more educational content. If you want more videos on specific topics in obstetrics and gynecology, reproductive medicine and surgery, embryology or gynae endoscopy or any chapter in any book, leave your suggestions in the comments. Remember, stay curious, keep learning, and take care. Thank you.